Under regulation of motor behavior, this is the second and final component in basal ganglia. We are looking at structure-function relationships and disorders. First of all, I'd like to draw your attention to the 5 important lecture objectives and the reading material, as you can see here. Let us recall that there is a reentrant circuitry from cortex back to cortex involving basal ganglia and thalamus. We may say that functionally speaking, we are indeed completing a loop from motivation to initiation and action in certain behaviors, motor and non-motor. We will talk about this when we address the so-called parallel loops or channels of circuitry going through basal ganglia. But generally speaking, there's always a piece of cortex which recruits a portion of striatum, and then the pallidum, a certain portion of thalamus, and then typically a similar or analogous area of cerebral cortex gets that reinforcement at the end. As a general rule, the striatum receives afferents, so it works as an input zone, and the pallidum sends out the efferents, typically to the thalamus. And it looks like this in a cross-section. Striatum receiving afferents, this would be putamen, and pallidum, in this case the internal segment of globus pallidus, sending the output. Having said that, I also recommend, if you need reviewing, you can watch this video here. We need to recognize that a large and very wide cortical area contributes to the afferents that go into the striatum. And specifically, there is an uneven distribution of these afferents. From somatosensory and somatomotor areas, the putamen receives most of the afferents, and from higher order and multimodal cortical areas, the afferents will go to the head of the caudate and the rest of the caudate. Now this in fact will play a role in what we call the multiple parallel loops or channels that we'll also address at the end of the lecture. But now let us look at the architecture of the striatum, the input receiving area, and it has been known since the late 1970s and early 80s that there are 2 main components in the striatum: the patch or striosome, these more isolated, patchy areas, and the much larger and confluent matrix. The matrix surrounds these patches, and the matrix is acetylcholinesterase rich. The striosomes or patches are acetylcholinesterase poor, but they are rich in peptides. We will spend most of our effort on describing what happens to the matrix, because the matrix is the one that receives the input from the sensory and motor cortex and then sends the output to globus pallidus. The striosomes or patches receive input from limbic areas of the cortex and project to substantia nigra. So the striosomes support basal ganglia function along these lines. We see the input from cortex. We are now in striatum, and we go to substantia nigra, feeding back to striatum. Nevertheless, most of our investigation will be spent in the matrix of the striatum, looking at what happens to the glutamatergic, the GABAergic, and dopaminergic influences. And here is our model of the striatal matrix, the cell types and neurochemistry. We are noticing that the medium spiny neuron makes up 95% of all striatal neurons. This will be our focal point. These medium spiny neurons are modulated by local interneurons, and of course they receiving afferents from far distant sources, so that they indeed integrate input from a variety of sources. Understand that the medium spiny neurons are projection neurons. Now, at the same time, acetylcholine interneurons, which make up 1 to 2% of all striatal neurons, these large aspiny neurons, local neurons, interneurons, cholinergic neurons will also make synapses on our main neuronal type. And likewise, GABA interneurons, which make up 4% of total cell population, also exist, such as here and here. And in these cases, GABA often colocalizes with various neuropeptides. Now, once again, it is very important to understand the variety of inputs and the sources of axon terminals impinging on this main neuronal type, which is to say that vast quantities of dopaminergic afferents are coming in from substantia nigra, pars compacta, for example. Of course, glutamatergic input is coming from cortex. Serotonergic input from raphe nuclei. That is not depicted here, but we imagine serotonergic input coming in. And the previously mentioned cholinergic fibers from interneurons. Now, all of these would be reflected in the nature and the distribution of neurotransmitter receptors on striatal neurons. The D1 and D2 dopamine receptors stand out. When we look at the dopaminergic category, and the nicotinic and particularly the muscarinic cholinergic receptors, when we look at the cholinergic categories. Of course, not surprisingly, the various types of glutamate receptors are also present here, and GABA receptors, GABA B and GABA A, and a wide array of serotonin receptors. You will delve into the study of serotonin receptors in neuropharmacology, but I can tell you right now that the 5-HT1 is an inhibitory type that is present on striatal neurons, and also some of these excitatory types. So the regulation by serotonin is, not surprisingly, quite complex. So with that, to summarize, the most important inputs to basal ganglia will go to striatum, and they come from cortex to caudate, cortex to putamen, the cortical ones, and also from substantia nigra. The one from substantia nigra contains dopamine from the pars compacta of substantia nigra going to striatum. So this is on the input side of things. How about the intrinsic organization of basal ganglia? We know that the signal goes from striatum to pallidum and then out. From striatum to pallidum and then out. We also know that the striatum is the main input and pallidum is the main output. We also know that the main cell type in striatum is the GABAergic medium spiny neuron. Well, that would be over here signaling to the next neuron in chain in globus pallidus. And even in the globus pallidus, the medium spiny neuron that I expect as output will be GABAergic. Very, very important to note that both of these neurons, the striatal ones and the pallidal ones, are GABAergic. Now, this suggests that a type of double inhibition, which is called disinhibition, will be a very important factor in the function of basal ganglia circuitry. How about the output? We have already discussed that the output of basal ganglia is primarily from globus pallidus, as highlighted here. First of all, the main output to thalamus is from the medial or internal segment or division of globus pallidus via ensal lenticularis and the lenticular fasciculus going to thalamus, to the VA and VL nuclei. Now from the lateral or external pallidal segment, the output goes to subthalamic nucleus. That uses the subthalamic fasciculus as a conduit. You may also recall that the substantia nigra pars reticulata over here has been considered as an analogous territory to globus pallidus. So it is, in a way, if you will, a displaced type of pallidum, the substantia nigra pars reticulata in midbrain. It has GABAergic neurons. It receives output from striatum, and its own output will go to superior colliculus and VL and VA nuclei of thalamus. Not surprisingly, these mechanisms will regulate eye movements, saccadic eye movements, contributing to the so-called oculomotor channel of basal ganglia looping neurocircuitry. Nevertheless, we have arrived at one of the most important slides in this presentation. We need to recognize that the signal originating from cerebral cortex can return to cortex via two distinct routes. One is the so-called direct pathway, stimulating the expression of motor programs, and the other one is the so-called indirect pathway, which does the opposite. It restrains the expression of motor and other behaviors, so they fulfill opposing functions. The direct pathway looks like this, from cortex to striatum to the internal or medial division of globus pallidus to thalamus and back to cortex. Direct pathway to allow the expression of motor programs. And then for the opposite, the indirect pathway from cortex to striatum to the external division of globus pallidus. Now we're using this extra station, the subthalamic nucleus. We're going back to internal division of pallidum and out to thalamus and now back to cortex, indirectly using this extra component that we identify as subthalamic nucleus to restrain movement. Now, in addition to that, there's also a so-called hyperdirect pathway going from cortex directly to subthalamic nucleus. Now, this is thought to work synergistically with the indirect pathway. But for the most part, we will focus on the direct and the indirect, such that we look at the direct pathways first. And again, the idea is that we're looking at movement initiation, understanding that under baseline conditions, at rest, the globus pallidus internal segment inhibits cortex through thalamus. There is a local and baseline tonic GABAergic tone from globus pallidus internal segment exerted on thalamus so that we're not allowing movement unless and until there is cortical activation, which comes down transiently when the premotor and sensory motor areas of the cortex are giving the command, so to speak, that we are getting ready to move. Now that is the movement from motivation to initiation, ultimately to action. And recognize that this occurs transiently and specifically. So the output of the cortex increases the firing rate of the putamen inhibitory neuron. The putamen inhibitory neuron, now transiently, inhibits the inhibitory neuron in globus pallidus, thereby disinhibiting thalamus. Look what happened. That initial inhibitory tone was taken away. So therefore, thalamus is released from inhibition, and now cortex, through thalamus, for example, the premotor area, we are back in cortex, the premotor area is activated. This mechanism, via the direct pathway, will allow the expression of motor programs and motor behaviors. There is gating in that sense, a permissive role in motor behaviors. On the role of dopamine in the striatum, we can say the following. The nigrostriatal dopaminergic pathway, of course, from pars compacta is the main source. It is feeding into striatum. There is a great modulatory role and tone from dopamine on the striatum, and it is of course exerted via the D1 and the D2 receptors. Now the D1 and D2 receptors are present on two separate sets of output neurons in the striatum. Of course, dopamine at the D1 receptor is excitatory, and dopamine at D2 is inhibitory. And the D1 receptor containing striatal neurons participate in the direct pathway, and the D2 receptor containing neurons are mainly in the indirect pathway, as indicated here. Which is to say that the net effect of dopamine on the striatum is typically going synergistically with the gating and the permissive role of basal ganglia on movement initiation. And this will be very clear in a disorder, for example, Parkinson's, where the dopamine tone is so greatly diminished. So with that, we have looked at the direct pathway. Now we're looking at the indirect striatal pathway using subthalamic nucleus. So once again, to sum up, ultimately, this will restrain the activation of cortex and restrain the expression of motor programs. But the way it occurs, that once again cortical activation is hitting neostriatum or dorsostriatum, but now neostriatum acts on the globus pallidus lateral segment or external segment, and that will go to subthalamic nucleus, essentially disinhibiting it. Remember, disinhibition will take place here and allow subthalamic nucleus to be released from inhibition. The signal goes back to globus pallidus, internal or medial division. What will that do? That can only increase the already existing negative GABAergic tone on thalamus. Remember, at rest, this was a tone that always existed, and now it became even more pronounced. Watch for the negative symbol to grow, and all of this is because of the disinhibition at the level of subthalamic nucleus. The role of dopamine, of course, is exerted via the D2 receptor, which has a negative influence, an inhibitory influence on neostriatum. So the bottom line is that the direct and indirect pathways work together. They may function to allow specific, meaning desired motor programs to go ahead, while at the same time other potentially confounding or competing motor programs are being inhibited by the indirect pathway. It is worth noting that the direct pathway receives a much more specific type of input from cortex, and the indirect pathway receives a more diffuse, broader input, not surprisingly, to selectively tune the output of thalamus that will go to cortex to specifically allow certain patterns of motor programs to take place. Which is to say also that diseases primarily affecting the indirect pathway will cause too much movement in that sense, unwanted movements breaking through, the so-called hyperkinetic manifestations. Balismus and choreiform disorders would be examples of this category. And for example, subthalamic nucleus damage, we are hovering here over the subthalamic nucleus, when it's damaged, will cause contralateral hemibalismus, flailing, ballistic, sudden limb movements. And in a similar way, degeneration of the D2 receptor bearing striatal neurons, many of them are in the caudate, but some in putamen, for example in Huntington's disease, will cause an awkward, dance-like choreiform type of movement. Also because it interferes with the normal ability of the indirect pathway to restrain unwanted movement behavior. This would be the review of functional circuitry. Obviously, it is a cortex to cortex kind of interaction going through basal ganglia in various ways. The first way is the direct stimulating motor programs, and the other way simultaneously is the so-called indirect using subthalamic nucleus and all the way back to cortex doing the opposite, which is to restrain movement so that normal movement, desired movement, is in fact initiated and competing and confounding other motor programs are simultaneously inhibited. We are at the point that we can look at some of our clinical highlights. It is fair to say that damage to basal ganglia is found in the broad category of movement disorders that neurologists recognize. And more specifically, of course, as I mentioned, hemiballismus is a sign of a contralateral subthalamic nucleus damage. And we know why. We're interfering with the normal functioning of the indirect pathway. Now, also in another way, and some of it is motor, some of it is non-motor, obsessive compulsive disorder and Tourette's syndrome are also thought to involve striatal dysfunction. And we will come back to this when we address the parallel channels through basal ganglia. It is also worth noting that, speaking of ventral basal ganglia, the ventral striatum and ventral pallidum, you remember nucleus accumbens and ventral pallidum, their work is related to the expression of mood, motivation, and they are strongly affected by drugs of abuse, particularly psychostimulants. Why? Because psychostimulants release dopamine. Now, neurologists will subdivide movement disorders into two opposing categories. At one end of the spectrum, there is slowness of movement and extreme difficulty initiating movement, and they are the bradykinetic disorders. At the other end of the spectrum, there are the hyperkinetic disorders. Now, speaking of the hypokinetic and bradykinetic, meaning slowness of movement disorders, Parkinsonism and Parkinson's disease will stand out. What is the difference? Parkinsonism and Parkinson's disease are both recognized by the diminished dopaminergic tone coming from substantia nigra, but Parkinsonism probably has a known cause, maybe from toxin or other related causes, whereas Parkinson's disease is idiopathic, meaning there is no known cause where there is degeneration of substantia nigra neurons. Now, speaking of the other syndromes here, some of these are the so-called atypical Parkinsonian syndromes, where there is Parkinsonism plus a whole bunch of other manifestations. And these, so for example, progressive supranuclear palsy, Lewy body dementia, and multiple system atrophy need to be, in fact, differentiated from idiopathic Parkinson's disease. So with that, the typical idiopathic Parkinson's disease is the second most common neurodegenerative condition after Alzheimer's disease. There is no definite cure for it. It was originally described by James Parkinson, a physician from London, in 1817, but it probably has been known for a very long time. Parkinson's disease affects approximately 8 million people worldwide, and it is estimated to affect about 1 million in the United States. The etiology, of course, is unknown. It was in 1960 when a scientist named Ole Hornykiewicz discovered that Parkinson's disease is in fact associated with the loss of dopamine input to the striatum. Starting this long revolution now in discoveries in neurochemistry and finer understanding of the brain. So nevertheless, observing these patients, the signs and symptoms will show you the typical presentation of a Parkinsonian patient. Bradykinesia, slowness of movement, and difficulty stopping. Once one gets started, it is also difficult to stop. Muscle rigidity, so-called cogwheel rigidity, sometimes the so-called lead pipe rigidity also occurs. Tremor, in many cases, not all the time, but in many cases. Of course, rotary, so-called pill-rolling tremor at rest. Postural instability, stooping posture is very, very typical. And the gait disturbance is evident in the observation of shuffling feet and difficulty starting and stopping. Cognitive impairment occurs usually late in the process, late in the development of the disease. So motor functions are compromised before cognitive functions are. As we now well know, Parkinson's disease is characterized by a severe deficiency in the nigrostriatal dopaminergic pathway. The cell loss in substantia nigra, the pars compacta, is evident. About 80% of pigmented neurons are lost from substantia nigra for the disease to be manifested clinically, and the remaining cells often show a Lewy body type of pathology. There are rare inherited cases with mutations, but in most cases it is sporadic and completely unknown why these cells die specifically, leading to the incredible loss of dopamine from neostriatum. So this was Parkinson's disease. In terms of hyperkinetic disorders, at the other end of the spectrum, fast components of abnormal movements, for example, athethosis or the choreiform movements and the ballistic movements of the limbs, but tics, myoclonus would also be listed. There is a faster component, and unwanted motor components break through, even at rest. The prototypical disease is Huntington's disease or Huntington's chorea. Strange, unwanted, dance-like movements of upper and lower limbs, head and neck, and mouth and face can appear. Usually there are behavioral and cognitive, depressive-like and irritability-like symptoms together or even preceding the motor manifestations. And it is, in fact, a heritable disease with a defective gene. This gene has too many repeats of the CAG trinucleotide. And the more repeats one has in this gene, it's called Huntington gene, the more severe and the earlier the presentation is. Nevertheless, neurons are lost, particularly in the head of the caudate nucleus, but also in putamen. So we see the normal brain here in an MRI, in an axial MRI, and then one with Huntington's disease, we see the enlarged ventricular system and the hollowing out of the frontal or anterior horn of the lateral ventricle here because of the complete, almost complete disappearance of the caudate head. A very typical presentation in Huntington's disease, of course, interfering with the normal functioning of the indirect pathway. So the indirect pathway is deficient here, stemming from the loss of the D2 receptor-bearing striatal neurons, causing an abnormal activation of cortex via thalamus. Once again, Huntington's disease, a prototypical form of a hyperkinetic disorder. We are at the point that we can discuss the series of parallel loops or functional channels going through basal ganglia circuitry. These are channels organized by areas of function. Each channel follows the same basic formula, but the components, whatever is recruited in this chain, are specific to the channel. There's always a cortical source of input, and that input will go to a certain part of the striatum, which identifies a certain component of pallidum that responds, and then the signal goes to the thalamus and back to cortex, typically to the same or similar area of the cortex from which the signal originated. But each channel differs in the identity of the specific areas that are being recruited for these functions. Which is to say that so far in this lecture, I have talked to you about the motor channel or the body movement channel via putamen, via the sensory motor areas of the cortex and premotor areas of the cortex, the putamen, the globus pallidus, the VA-VL nuclei of thalamus, and back to these same areas of the cortex. So far, we have really focused on the motor or body movement channel. I also mentioned the oculomotor channel, if you recall. Of course, that's originating from this area of the cortex. The oculomotor channel will go to caudate for the most part, and then we'll go to the substantia nigra pars reticulata, and then to thalamus, and back to cortex, and some of it will go through superior colliculus, as I mentioned. That is the oculomotor channel. In addition to these, we also have the prefrontal or cognitive channel that originates from this broad lateral and dorsolateral area of prefrontal cortex and will signal through caudate for the most part, will signal through other areas of the thalamus, such as dorsomedial nucleus, and then will go back to the same area of the cortex. And the fourth one is our limbic channel. The limbic channel originates from limbic areas of the cortex and the amygdala and hippocampus. And this is the one that engages the ventral striatal pathway and the ventral pallidum pathway, going to thalamus and going back to the same limbic areas of cerebral cortex. Please recognize that the first three are dorsal striatal patterns and pathways because of their localization, but then the last two, prefrontal and the limbic channels, functionally are the non-motor loops. Why is that? Because the prefrontal or cognitive loop and the limbic loop, they regulate gating of functions in planning, attention, short-term memory, emotional, and motivated behaviors. So they are non-motor primarily, and some of them may overlap with what you might call psychomotor functions and initiative taking in a much broader sense now. So having said that, let us focus on the limbic loop that we recognize as the one that goes through ventral basal ganglia. And of course, the significance of it is given here because it's related to drive and mood types of functions, and indeed regulates the relationship between stimuli and rewards. It gets its dopamine supply primarily not from substantia nigra, but the adjacent ventral tegmental area. And of course, it starts in limbic cortex, hippocampus, and amygdala, and then ventral striatum, or nucleus accumbens, is recruited, and then VP here, ventral pallidum, is recruited, and then the signal goes through dorsomedial nucleus of the thalamus to be fed back to the same or similar areas of cerebral cortex. So once again, this was the limbic loop, specifically signaling via the ventral basal ganglia, using dopamine from ventral tegmental area. At the end, I would like to summarize the significance of parallel channels or loops more from a clinical point of view, as dysfunctions of these are manifested in clinical presentations of various types of diseases. And of course, we are not at all surprised that Parkinson's disease primarily affects the motor loop. And of course, primarily the direct pathway through putamen and the rest. Huntington's disease affects the motor and the prefrontal loops. Again, not surprisingly, there is more interaction from the head of the caudate, which is linked to prefrontal cortical areas and higher order areas, plus the motor manifestations. It is rather mixed. Huntington's, motor, and prefrontal. And now we're looking at Tourette's syndrome and obsessive disorders. Of course, Tourette's syndrome has vocal tics, motor and complex tics, various types of obsessive behaviors, and then obsessive behaviors themselves. may involve a dysfunction in the prefrontal and limbic channels, and may also involve an abnormal activation of orbital frontal cortex. So now we're moving to that ventral area of prefrontal cortex to evoke the elaborate motor patterns as compulsions. But of course, the emotional processing at the same time is also disordered, not surprisingly. And now for depression, on the one hand, mania at the other extreme in that functional spectrum, and in cases of drugs of abuse, they have a tendency of targeting the limbic and prefrontal channels in various combinations. And we say various combinations because that will depend on clinical presentation, not surprisingly, whether a hypo or hyperactivity of the cerebral cortex is postulated. And indeed, some cases it would be a disordered gating as far as being too permissive, or on the other hand, too restrictive in that sense. Very much in line with the general principles that we have learned about basal ganglia overall. So with that, this was our second and final installation of the basal ganglia topic, having looked at cell types, neurochemistry, and some important examples of pathologies. So with that, thank you for your attention.